Hello and welcome to another episode of the IBMS podcast. I'm Rob Dabrowski, the editor of the Biomedical Scientist magazine, and in this instalment I'm joined by Richard Burka, a Haematology Registrar and academic clinical lecturer. Our discussion covers a range of areas from anti-coagulant reversal agents and the importance of randomised control trials through to the power of storytelling. I hope you enjoy the episode. Hello and thanks for joining us today. I'm also joined by Richard Burka, who I'm going to pass you over to straight away to introduce himself. Richard, do you want to tell us a little bit about yourself and your background and your current roles?
Hi Rob, yeah, sure. Thanks for having me on. We met at the recent IBMS Congress in Birmingham, which I was almost catastrophically late to for a 9 a.m. lecture. So we got there in the end. But I was asked to speak at that, and we'll touch on why. So I'm an academic clinical lecturer, an SD7 Haematology Registrar in Birmingham. I've been in and out of research jobs for best part of 10 years, starting with integration at med school in cancer immunology. Gave me a bit of a bug for the lab, the discovery science side of things, and then rekindled all that as an academic foundation program. Did a bit of epidemiology work along the way, real world evidence type stuff, and then applied for a couple of clinical research training fellowships, so sort of clinical PhD fellowships, which are pretty hard to get. I applied 3 times with one project and failed miserably with all of them. And then got to know Pip Nicholson, who's a consultant haematologist in Birmingham, and we fell onto a project on reversal of anticoagulation. So we collected data nationally on that, and then Pip said, "Well, let's try and turn this into a PhD." So I applied for a British Heart Foundation accelerator, which was administered by the University of Birmingham, with a project on reversal of anticoagulation, what that does to thromboinflammation. So collecting samples of patients who have been reversed with andexanet or PCC or whatever, and looking in the blood for markers of inflammation. So they liked me, I think, and gave me the job. And on day one, our professor in Birmingham, Steve Watson, says, "I think you were great, but the project's really terrible. Why don't you do this instead?" And it was 2022. We're just coming out of COVID, and the big story in thrombosis was VIT. So this is vaccine-induced immune thrombocytopenia with thrombosis, or variations of that terminology. So this is post-COVID vaccine blood clots. And we had 12 patients in Birmingham with this disease, which I can talk about if anyone's interested, that we then interrogated their blood samples and did some discovery science in the lab. And that's what I did for 3 years ending last February. Really loved that experience, really loved the PhD. And now I am a NIHR clinical lecturer. So I do half research and half clinical, although I'm 80% less than full time because I've got 3 kids under 5 and I live about an hour and a half away from work and life is tough. So it just buys me a bit more time to do various bits. But interesting, I looked at the less than full time contract, it's still over 40 hours a week and the rest of everything else. And then my final hat, I guess, is I'm chair of Haemstar, which is a UK-wide network for haematology registrars and others interested in medical haematology. So that's all of haematology that's not cancer, and we do sort of projects and audits and surveys in that field and try to support people to have academic interests and careers in that area. So that's me really, keeping busy.
Are you all done with your PhD now, Richard? Is that
Finished and awarded, happily.
And how long did that take you? Did you say it was 3 years, which seems pretty speedy for a PhD these days?
Yeah, from start to finish, 3 years. I started February 22, finished in the lab September 2024, so 2.5 years in the lab basically, 4 or 5 months writing up, then back into clinical medicine for 5 or 6 months. Did my FIVA last Easter, and then the graduation was July.
I find it quite interesting that you went in with one kind of particular topic and then they were like, nah, COVID's kind of changed everything. And it seems like all the medical funding after COVID was just kind of swallowed up and pumped into anything that was vaguely related to COVID. I mean, do you think that the work you were originally going to do would have been interesting and it was just the kind of circumstances of COVID that changed the focus of medical research and funding? Or were you fundamentally looking at something that was a bit rubbish and you were like, oh, thank God we've changed it to something else?
Yeah, I think it's more the latter. Steve Watson's very experienced, and I think he decided my project as stamp collecting, which is essentially for the sake of it getting data, but it's not particularly hypothesis-led. So the VIT project was such a better project. Interesting, that stemmed from them having some national funding. So when VIT happened, Chris Whitty, etc., put together a consortium of researchers around the country with expertise in thrombosis and epidemiology. So there was a consortium with 5 work packages involving people in genetics, people in Public Health England, people like us in thromboinflammation, and we in Birmingham were a platelet activation group. So we look at platelet signaling basically. And actually a lot of that work has continued since I finished, with my colleague and PhD supervisor Sam Montague working on various aspects of that. So there was quite a lot of funding for VIT. And I think the guys, so this was led by Professor Munir Pir Mohammed, who was, I think, president of the British Pharmacology Association. I might have got that wrong, I apologise. But very eminent, brilliant, excellent at getting money and holding together a very, very cohesive group, which actually developed in quite a nice community. But the argument was that it's really important to understand VIT, not just because of the 300 or so patients that were affected and trying to understand what happened, but also the fact that adenoviral vector vaccine, that platform is exceptionally powerful, exceptionally agile, and it is not beyond the realms of possibility that we have another pandemic in the next 5 or 10 years. And yes, we have mRNA vaccines, but the adenoviral vector vaccine is such a powerful platform, and it can be repurposed so quickly that, as we saw in COVID, even though we saw 1 in 50 to 1 in 100,000 people develop VIT, that vaccine saved countless lives. Now, that's not to belittle the experience of people who had VIT, which is horrific. And we know that people are still suffering, but you can't get away from the fact that those vaccines saved millions of people worldwide.
Can you talk us through VIT a little bit and what exactly is it? My knowledge is limited.
Yeah, sure. So this is a disease that's related to heparin-induced thrombocytopenia, which some may have a bit more familiarity about. So in a nutshell, and interestingly, if you're interested, there is an In a Nutshell article for BJ Heme that I wrote with Sue Pavid. In a nutshell, these diseases center on a molecule called platelet factor 4. So PF4 is a fascinating molecule. It's the most abundant molecule in platelet granules, making around 2.5% of platelet granule proteins. So it's extruded from cells when platelets get activated. And what it is very positively charged. So it's like a sphere, it's like a globe. And around the equatorial part, you've got a positively charged ring, positively charged amino acids. Now, in HIT, heparin binds to that. So heparin is one of the most negatively charged molecules in biology. Heparin will bind to PF4, and it will overcome the positive charge, allowing those PF4 molecules to cluster together, and they form these large immune complexes. Now, what that does is that when heparin binds to PF4, there's an energy shift. It changes the conformation of platelet factor 4. It opens it up and it exposes neoepitopes. It exposes areas that antibodies can bind. So you end up with an immune complex that's made up of PF4, heparin, and antibodies. And antibodies are Y-shaped proteins with the top of the Y facing towards the PF4, and sticking up or sticking down, whichever way you want to go, is the FC region. It's that FC region that binds to receptors on platelets and white cells, activates them, and causes blood clotting, consumption of platelets, and in some cases bleeding. So that's HIT, that's heparin-induced thrombocytopenia. We see this as a consequence of giving people heparin therapy, but actually you see these antibodies come up in infection, in severe COVID, and even if you go and look in healthy blood donors, you can see anti-PF4 heparin antibodies. And it's thought that that's because of an evolutionarily conserved immune response, because the walls of bacteria are strongly negatively charged, and the immune system can't otherwise see negative charge as a danger signal. So it's evolved this amazing system where you use a very positively charged endogenous protein, your own body's protein. It goes and binds to the negatively charged surface of the bacterium, changes conformation, and then an antibody can see that change in structure. So it's an amazing way of bridging innate and adaptive immunity by converting charge into structure. So PF4 acting as a bridge to convert charge into structure. So that's heparin-induced thrombocytopenia, and that's the evolution. How is that different to VIT? Well, in VIT, instead of needing heparin, the antibodies bind to the equatorial ring of positive charge, and they cluster the PF4 molecules on their own. They're very powerful, strong antibodies that can themselves overcome that strong positive charge. And we think that's pretty rare. There's a few alleles, there's a few immunological there's one particular IgG allele that predisposes people to VIT, but that's present in around 20% of the population, and only 1 in 50,000 had VIT. So something else happens, and we don't really know why yet, although work's ongoing on that.
What are the kind of consequences of having VIT when you see someone with it? How are they?
So the most intriguing thing is that around half of people who have VIT presented with cerebral venous sinus thrombosis. So this is a blood clot in the veins of the brain, which is otherwise a pretty rare site for thrombosis. And that was the most striking weirdness. And that doesn't happen in HIT. So there's something different going on. Although you've got very similar pathophysiology, you don't see CVST, cerebral venous sinus thrombosis, in HIT. You only really see it in VIT. And again, we don't really know why that is. So we see thrombosis, we see low platelet counts, and if untreated or poorly treated, it's got a very, very high mortality. In the UK, I think a third of our patients with VIT died. Interestingly, as it became recognised and people worked out what on earth was going on and started using things like IVIG and plasma exchange and steroids early, the mortality fell. So contrasting our outcomes with Australian outcomes, and they were about 4 weeks behind in their vaccination program. So by the time we started seeing VIT cases, they hadn't seen many. And by the time we'd recognised what it was, they were able to leverage that useful information and treat their patients more aggressively because they knew what was going on. So hallmarks of VIT: thrombosis, thrombocytopenia, and a generally very unwell patient. You see very high D-dimers, you see very low fibrinogen, and low platelet counts.
And you say we don't really know the exact reasons why it happens. Is that something that we can find out with research, or is it rare enough, so that there's no one wanting to put funding into it, there aren't enough people looking at the problem? Is it something we can find out what's going on, or is that unlikely?
There's a lot of things going on worldwide. So there are groups in Canada, Australia, and Germany who are a long way ahead of us. They've been working on these diseases for a long time, and I know that work is there is this information is coming. It is coming.
Nice. That's good news. Let's loop back to Congress, Richard, where I met you, and you did a fascinating talk on anti-coagulant reversal. This was related to your PhD, right?
So in a way, I guess. So because I applied for the PhD in Birmingham with this reversal project and then started, it's a long story, but it's quite hard to get funding. So my funding for the PhD was quite bitty. Some of it came from the NIHR grant on VIT. Some of it came from the British Heart Foundation Accelerator, and some of it came from an additional BHF, British Heart Foundation funding stream. But so I started on the VIT grant with the intention of then going back to my project on reversal. But we found some interesting stuff in the first 6-7 months. And I sort of said, well, I'll just carry on doing the VIT stuff, and left the science on DOAC reversal behind, because I think it would have been really challenging to show anything meaningful. But we'd already set in motion a national project on the clinical outcome. So a national audit looking at patients who receive reversal agents with direct oral anticoagulants. So this is apixaban, rivaroxaban, edoxaban, dabigatran, those 4 drugs. So that was called RAPIDO. And I can never remember what that stands for. It's like reversal of anticoagulated patients on DOAC or something. And we've gathered nearly 2,500 patients worth of data on that. The first manuscript's looking at just the bleeding patients. There's a second follow-up looking at patients who had reversal agents prior to urgent surgery. And the first manuscript's been accepted into JTH, so that should come out very soon. And we're quite excited about these findings. Look, it's a real-world evidence audit, so it's never going to prove very much. But what we were able to do is really assess how these drugs are used, where the problems are, and generate some hypotheses. And that's what I'm working on now. The thing that's interesting, I think, that's come out of this, and from looking at the trials of reversal agents, and I can touch on what these reversal agents are and where we are, but the most interesting thing for me is that, one, I think we have evidence of overuse of these drugs that are quite powerful. They have problems, they have side effects, they can cause blood clots on their own, and there's no real good evidence that they improve patient outcomes. So one, we've got a little bit of evidence of overuse, but secondly, we've got evidence that they're used quite late. So in patients who have apixaban or rivaroxaban, these drugs wear off quite quickly. So by the time they hit hospital with a bleed, often the blood thinner's wearing off. And we know in the trials, there's 2 big trials of a reversal agent called andexanet alpha. They use a cutoff for inclusion in those trials of either 15 or 18 hours following the last dose of anticoagulant. So for example, rivaroxaban, you take it once a day. If you take it at 8 o'clock and you come into hospital at, let's say, 11 p.m., you'll be excluded from the trial. Is that right? Oh, my maths is shocking. No, not quite, but you see what I mean. So if you come in 15 hours after you've taken, you'll be excluded from the trial. And we know in those trials, even without exclusion, around 25% of people have a subtherapeutic level. So you think, well, actually, we can try and exclude those people from having these powerful clotting agents, these powerful drugs. We can save money. We can prevent thrombosis by having access to urgent DOAC level testing. But we just don't have it. So there's evidence internationally that these kind of pathways can work, but across the UK, unless any of your listeners know differently to me, I don't think there's anyone that's really got urgent DOAC level testing up and running.
Sorry to jump in, Richard. Are there other countries that do, or is it something that's not really known?
So I don't really know. I've got a survey that we've been working on that's hopefully going to find out some of this information that we're going to send internationally. My suspicion is that, yes, it's recommended in guidelines that you should do levels, but my suspicion is that there's a lot of reluctance because people worry about the time it takes. So people worry, you've got a bleeding patient in front of you. You don't have half an hour, 45 minutes to wait for a level to give a potentially life-saving therapy. My argument is that we don't have evidence that that it is a life-saving therapy. And actually, I've been gathering some local data that shows there is time. So even if people say this is urgent, rarely are people given a reversal agent within 2 or 3 hours of hitting hospital. It's usually a lot longer than that. There's a lot of delays. And actually, if you've got a drug level being tested in the background while those delays are ongoing, then that doesn't slow anything down. So my argument is actually, okay, people say, okay, Rich, there's not time. My argument is actually the data shows there is.
And how easy would it be to set up this testing?
So this is the tricky bit. So the testing exists. So my proposal is you would do a anti-10A factor activity assay, which is either clotting based or chromogenic based. All big labs have the machines available to do those. You just need the will and the calibration and sort of the staffing to do it. And you need to upskill your staff to be able to do it. Now, one of the problems is that you need specific calibration for different drugs. So if you want to know what the apixaban level is, for example, you need a apixaban calibrated machine. So if you've got patients that are coming in, one's apixaban, one was rivaroxaban, one's adoxaban, like there's some difficulty there. What you can do though is you can do a low molecular weight heparin calibrated anti-factor 10A assay. I don't know, that's a mouthful. And it won't give you a reliable quantitative result. But what it will do is it will tell you definitively, if you use a particular cutoff, that your patient is definitely under a certain level. So we're interested in patients under a level of probably 50 nanograms per mil or maybe 30 nanograms per mil. But on a low molecular weight heparin calibrated anti-factor 10A activity, if we use a cutoff of 30, we can pretty much guarantee that the patient's under 50. Mm-hmm. Well, your question is, how feasible is this? Well, the technology exists. We have the machines. We have these assays up and running. It's the real world human bit that we need. And I think it comes down to the story. It's such a powerful story to want to reverse someone. It's such a powerful story to give someone an antidote to a blood thinner that we think's made them bleed. But unlike with warfarin, where it hangs around for a long, long time, as I've said, these drugs wear off quite quickly. They're out the system. And there's other kind of biological things where you think, well, maybe it doesn't impact bleeding as much as warfarin maybe does. The rationale for actually reversing people is much, much weaker in my mind. And again, we don't have definitive evidence. Despite how logical it might be, despite how intuitive it might be, despite how good the story is, we don't have definitive evidence that giving any reversal agent to anyone who's bleeding on a DOAC changes their outcome. In fact, we've got a randomised controlled trial in people with intracerebral hemorrhage that randomised patients to andexanet versus standard of care. A significant number of patients who received andexanet had a reduction in their hematoma expansion over 12 hours, so how much the bleed grew. But that had no relation to clinical outcomes. There was no difference in disability or death. There was no difference in quality of life, not that that was measured at 30 days. So there's some real deficiencies in the evidence. And what's more, that drug causes stroke. So we think that it doubles the risk of stroke post reversal. So all in all, it's risky. We know there's definitely evidence of harm, and there's no good evidence of benefit.
So were you surprised when you discovered that these decisions were being made not on an evidence base and not on outcomes, but on essentially a narrative and people wanting to take action? Was that a big surprise, or did you suspect that when you were starting this work?
So I'm not surprised. When we started the work, we knew there was no good evidence. So we knew that it was based on story. I think over the last 5 years or so, I've been really interested in the story side of medicine and what we tell ourselves and how powerful stories are. Interestingly, there's a now it's in Nature Evolutionary Medicine or something. There's a paper from a while ago showing that if you're a good storyteller, you're more evolutionarily successful. So the way we understand the world is through stories. We understand our own lives through our own story and what we tell us. So story is exceptionally powerful. And the thing with reversal, I think, is that because it's so intuitive that someone's bleeding, they're on a blood thinner, we must reverse them. That is a really intuitive, logical thing to do, that it's really difficult to fight against that. I mean, randomization itself is not something that's inherently intuitive for humans. The first randomized trial, proper randomized trial, and we'll discount James Lind in this 18th century. Well, I'll come back to that. First proper randomized trial wasn't until 1950s of streptomycin in TB. And the MRC only did that because there was a limited amount of streptomycin coming into the UK, and they thought, well, how do we give this away equitably? So they just sort of randomly did it. So that was the first proper modernised randomised controlled trial. Some of the listeners might know about James Lind, who was a British Navy surgeon in the 18th century. I forget the date. But he was interested in scurvy. At that time, they didn't know what it was. And he did a trial where he took 12 sailors and gave 2 sailors one treatment, 2 sailors another treatment, 2 sailors another treatment, one of those treatments being citrus fruit, and observed that the guys that got citrus fruit totally resolved their symptoms of scurvy. But even he couldn't really understand the implications of his findings. He sort of really just discounted it all. And it wasn't until many, many decades later that the British Navy actually instituted this treatment. So there's a reason why it's taken humans 300,000 years of evolution to come up with randomization. And the reason is that it's not intuitive. In fact, I think it's one of the greatest leaps in human science ever. Randomization is so powerful and so simple, but just goes against our psychology, because what we want to do is believe our lived experience.
Mm-hmm.
So what we really need is, I don't think it's reasonable for me to say to people, right, there's no evidence for reversal, you must stop doing it, because that's not going to happen. The ship sailed, there's a tidal wave, I can't hold that back. So what we need to do is we need better evidence to allow people to be rational, even where it feels unreasonable. That's my speech for you.
It's a good speech. To ask you to cast your kind of net over a massively broad range, Rich, I mean, do you think there are many other areas in medicine where we have similar things, where there's an assumption being made without an evidence base because there's a great story? And is this one of those where in, 100 years' time, people are going to be looking back and saying, why on earth were they doing all these idiotic things without any evidence when, this is what we do now? In a similar way, we look back at the medicine of the past and think, isn't it ridiculous that they did that? But they obviously at the time thought that was the best thing to do. Otherwise, they wouldn't have done it.
Yeah, I absolutely think that's possible. And that's the case. I think there's 2 things I'll say. One, there's a lot of writing on something called medical reversal. And this is where intuitive practices are widespread. And then someone does a trial to say, actually, this doesn't work. So actually, a very recent example, patients with atrial fibrillation, we know that most blood clots, if you've got AF, come from the left atrial appendage, which is basically a little pocket on the left atrium. So one of the treatments for that has been to do a left atrial appendage occlusion, so close that occlusion with a device. And the patients that people have preferred to do that on are ones where they've got AF, we know they're at high risk of stroke, but they also have a high risk of bleeding. So they have a contraindication to an anticoagulant. So you don't really want to give them an anticoagulant. So instead you go and close this thing. But there's actually a trial very recently where they randomized patients to standard of care versus left atrial appendage occlusion plus aspirin. So most of the standard of care were actually put on a DOAC. So how we're represented, I don't know. But it shows actually a higher risk in the ones who had the left atrial appendage occlusion closure. High risk of bleeding, which is counterintuitive, probably because of aspirin. So there's a very recent example of something that's widespread, people do all the time, it's intuitive, but actually when you go and test it, it makes a difference. So in my opinion, the only time that you don't need a randomized trial, ideally, is where something's a medical parachute. So a parachute is where, you jump out of a plane and you've got 100% risk of death unless you have a parachute. And the parachute changes. So vitamin C for scurvy, for example, is a medical parachute. It cures everyone. And it's basically got no risk. If you don't have a medical parachute, then you need to test it. So to me, direct reversal, although very intuitive, numbers needed to treat are not one. So a parachute is defined as a number needed to treat of one. You need to treat one person to save one life. At best, and we don't really have the data, but at best, direct reversal number needed to treat to prevent one significant hematoma expansion, around 14, and you're causing stroke as well. So there's a number needed to harm there. So I think that's important. I think randomized trials are important. The only thing I would say against that is that there are diseases like myeloma. So this is going off non-malignant hematology, but diseases like myeloma, there have been randomized control trials. They've not been perfect. But clearly, the progress we've made over the last 20 years is massive because you can just see the benefit in survival. So you can't just do perfect randomized control trials all the time. But what I think we should do is we take the things that are expensive or harmful, potentially harmful, and really question the dogma of whether they're right or not and test them. Unfortunately, sometimes there's not funding to do that because, a lot of the funding for trials and stuff comes from pharmaceutical companies or device companies where there's skin in the game and, the purpose of the trial really is to sell the drug. But we as academics need to try and campaign for better evidence, particularly when there's good evidence of harm or a lot of financial toxicity.
Is it a lot harder to get a randomized control trial in place for something that's already being done than something that's coming up? For example, some new drugs coming along, randomized control trial. But is it a lot more tricky when there's already an established kind of process in place?
I think there's two reasons it's difficult. One is funding. So if I've got an approved drug that's been put through trials by the drug company, have paid for the trial, then it's very difficult to do anything to stop that. And the second thing is the story again. So to do a randomized trial, you've got to have patients and doctors who are willing to randomize and be randomized. And if there's a powerful narrative already, it's very difficult to convince people to do that.
Yeah. There's a guy who used to write in The Guardian, did a column called Bad Science, Ben Goldacre. And I remember reading something from him, and sorry to him if I'm miscrediting this, but he was saying that he thought the results of all trials should be published somewhere, regardless of what they said. Because, say, for example, you've got a drug company, they want to sell a new drug, they try it out 20 times in trials, it shows no difference, and then they have one trial and it does show a difference, and that's the one they publish. Do we need not only more randomized control trials, but more publishing of kind of everything that's going on in research, or is that just going to create kind of a flood of information that people can't process?
Look, I'm always up for transparency. I think trials have improved. So there are prospective trials registers now that demand publication, but you definitely see it that if a drug company produces a positive trial and there's a positive press release, the trial comes out quickly. If it's a negative trial, as we've seen with crizanlizumab for sickle cell disease, definitely there was a phase 3 study that was essentially negative. That took ages to come out. So there's definitely delays. I mean, Prof. Goldacre is the guy to ask on this. I don't know the literature enough to know how things have changed or whether they've improved or not. There's a wider question about publishing of negative results. There's definitely bias towards publishing positive results because, again, it's a better story. And if you have positive results, then it's easier to publish in a good journal because, again, it's a good story. It sells the journal. People cite it. But negative results are important too. I went to a great talk from one of our colleagues in Birmingham the other day who talked about negative data and actually how they completely refuted another group's work. It took them a long time to get it published. It was pretty stressful to try and disprove something. And he said it was worth it. He said probably if it was 10 years ago, he probably wouldn't have done it from a career point of view. But he said it's established now, doesn't care. But it takes people to take a stand. And there's a lot of science is not reproducible. So there's data on this, and I can't remember can't remember the studies, but there's data on exactly how reproducible things are. Some work from John Ioannidis, who is along in the sort of same ilk as Ben Goldacre, publishing this kind of stuff, looking at reproducibility, and it is a real problem. And we know that academic fraud's a real problem. Randomization gets over some things, but there have been examples of trials where people have just made up the patients. You can't mitigate against that. It's difficult. It's difficult. But I definitely work on people looking through studies and working out what's totally implausible and what can't be correct. And that's actually pretty widespread.
And at the moment, are you a haematology registrar? Is that right, Richard? Is that...
Yeah.
So that means you're training to be a consultant haematologist?
Yeah, I'm a little bit different because I've done a lot of sort of research on the side. So it's a bit of a circuitous route. It's taken me a little while. So yeah, I'm SC7 at the minute. So I'll finish off training end of next year. I've got to do my FRC Path Part 2 exam in the middle somewhere, which is a bit of a hurdle. But yeah, nearly there as a consultant. But I'm also a clinical lecturer, so that gives me half and half research time.
And once you're there, is that kind of a dual role in the lab and kind of patient-facing as well?
So my aim would be to be an academic clinician with a split. So something like a 50-50 or a 40-60 or a 20-80 split. So I always want to do research. I think it's what gets me up in the morning. Again, like there's a story I'm trying to tell myself, I suppose, that I can make a difference. It just gives me a lot of interest and enjoyment, and conversations like this and going to interesting conferences and collaborations with colleagues. And you mentioned Ben Goldacre, I've collaborated with his team on stuff recently. So I think you can make a real difference through research. And I think I can make a difference by banging the drum for better evidence and telling a story better.
Yeah. And I think you're an incredibly good storyteller yourself, Richard, and very good putting across information with some incredibly long words and some complex theories that is kind of consumable to a normal person and not just kind of, the top level academia. Can you tell us a little bit about, you've got a podcast. Is it Don't Just Read the Abstract, it's called?
That's right. Yeah, so Don't Just Read the Abstract, I co-host with my friend and colleague Pip Nicholson, who supervised my PhD, supported me for many years, and I just love doing that with Pip. He's very meticulous. He contrasts with me really, really nicely. I try to wing it a little bit, and Pip does his homework. So that's amazing. I started a podcast a few years ago called Don't Just Read the Guidelines, where I started interviewing some interesting people, up and coming scientists. I just kind of wanted to give a platform for people who were more up and coming, like me, I guess, and who had good stories to tell and good opinions. But after a while, it gets kind of dry doing on your own. And I got to know Pip, and we applied for a little bit of funding to start the podcast and got that. And that went into Haemstar as well. So that was good. And so, we try and do a podcast every couple of weeks, every month or so, really trying to fill a niche in medical haematology about critical appraisal. So there's quite a few podcasts internationally that look at cancer, malignant haematology, but nothing that we really knew of that covered the whole gamut of non-malignant haematology. So we'll do anything from red cells, hemoglobinopathies, transfusion, hemostasis, thrombosis, or immunohematology. We try and critique, we try and interview study authors, and we do the odd kind of other interesting interviews, like one of our colleagues from Sheffield who spent a couple of years working as the only haematologist in Gambia. That's an interesting one for lab people, actually. Her perspective on how the lab works in the Gambia, and literally going to the medical director and him giving her cash to go out and buy reagents. Things we take for granted in the UK, things like INR reagents that just don't have or don't necessarily have access to. So if you want to go back and look at that one, that's with Megan Kell and Don't Just Read the Abstract. But yeah, we love it. Pip uses it as his CPD, actually. Yeah. So it sort of kills two birds with one stone. But we hope that it inspires other people to really read. And the name is indicative of what we're trying to do. The story that drug companies will try and tell you about drugs is not always the same as what the reality of the data is. So you have to read beyond the abstract. You have to critique stuff before you let it change your practice.
Nice. And what kind of format, just so people can get an idea of it if they want to go and have a listen? Is it you and Pip having a chat and then you bring in a guest and you discuss a kind of topical issue?
Yeah, it's mostly just Pip and I having a ramble about paper. We initially started quite ambitiously thinking, okay, well, we'll do a little news section, then we'll discuss a paper between us, then we'll have an interview with an expert, and then we'll have an interview with a study author. We realised it was far too much to do in one. So what we tend to do is do a full episode on critique. We do always do a bit of news at the start, so of the papers that we've seen or conferences or interesting cases we've seen. And then we spend about 45 minutes critiquing a paper. And then if we can, we'd like to get study authors on. Sometimes they say yes, sometimes they say no. If we can't, we often get an expert on. So there's some ones we've done on reversal where we've discussed the NXRI study of Index and Alpha. And then on the second episode of that, we interviewed Will Lester, who's one of our consultant colleagues in Birmingham and chair of the BSH Hemostasis Thrombosis Task Force. So a good guy to have on and a good perspective. So yeah, we mix it up a little bit.
Cool. And you do a blog as well, is that right?
So we've got a website called don'tjustreadtheabstract.com. Which also has a newsletter. So we started we started a twice monthly newsletter that we quickly realised was unmanageable. So it's dropped to a monthly newsletter, but it's quite clinical. The idea is to try and give people updates and news in medical haematology. Again, trying to fill a niche really, because there's quite a lot going on in malignant haematology and non-malignant gets short shrift. So we try to bring a couple of interesting news articles, share a couple of interesting publications, talk about what we've done on the podcast, a list of any upcoming events, and a few other nuggets of things like things we're reading or whatnot. Interestingly, one of the newsletters was I was reading the Science of Storytelling by Will Storr. So I put that in the podcast, in the newsletter, which is an interesting book if anyone's interested.
One of the main things that we keep on touching on throughout this chat is the kind of the art of storytelling and the power it can have. And obviously, the Institute of Biomedical Science, and before the pandemic, a lot of people weren't that familiar with a biomedical scientist, even people who work in the medical profession. And I feel like because they've been able to tell they've had a reason to tell their story, and people have been wanting to know how the diagnostics of COVID worked and what was happening in the labs and the difference between lateral flow tests and different types of tests. I mean, do you think that a sector of science can kind of use storytelling to progress their cause further? Because I think personally, I think that biomedical scientists have been doing that quite well over the last few years.
I think everyone has to. I think if you want to build something in life, you have to tell the story. You look at companies, there's a story around every company. What's John Lewis's story? It said, we do a great Christmas ad every year, we look after our members, our staff have I'm not a John Lewis shareholder, but we look after our staff. There's a story around that company, isn't there? There's a story around Coca-Cola, there's a story around cost, all these different companies. And it's the same. It's the same with professional organizations. There has to be a story. That story can be spun from the top. I think this is one of the failures of our government at the moment, actually. And I think that they're just not telling the counter narrative well enough. I don't want to get into the rest of politics or anything, but there's a very strong, powerful right-wing narrative about immigration and all these things. But I don't feel like there's a strong counter narrative of all the advantages that having a progressive left of centre government that makes responsible decisions can do.
Mm-hmm.
It's harder to tell that narrative because it's more complex, but we have to try and work towards that. Going back to the IBMS, we know biomedical scientists are massively important. We wouldn't be doing this podcast unless we really respected that job role. I guess the organisation needs to ask, firstly, what's the point of telling a story? Is it to attract amazing talent? Is it to attract more funding? Is it to, I don't know what else. What's the point? And then what is that story? When I walk through the hospital lab, for me, the story is, look at these people working away, actually removed from the patients, so they don't necessarily see that. But look how dedicated they are at doing this and striving for excellence. Look how diverse we are. Look how busy we are. And look how much of a difference we can make. And I think painting the story of how much the lab can make a difference is important. And if I go back to what I want to do with the diet reversal and the levels, there's a really powerful story there. And the top line of that is we can save money, we can prevent thrombosis, but we just need the will in the lab to do that. And I think that's powerful. You have to tell that story. But I think going back to the IBMS, it's just harnessing what the point of telling the story is.
Mm-hmm. Perfect. Richard, I think that's the perfect moment for us to leave it on. Really, really nice note to end on. Where can people find you online, just so they don't have to rewind and go through and look it up again? If people want to find your podcast, where should they go?
Some shameless plugs. So you can go to don'tjustreadtheabstract.com, which is our hosting website. You can find the podcast on all major podcast platforms, Spotify, Apple, wherever you get your podcasts. Sign up for the newsletter, as I say, at don'tjustreadtheabstract.com. I'm on LinkedIn, X, and you can find my university page if you just Google me. But I'm more than happy to hear from people. And again, if there are people around the country who are really interested in being involved in a project going forwards to try and look at levels, and my plan is to do a prospective trial on this, to really look and investigate the impact of putting urgent levels in a clinical workflow. If people are interested in that, just please contact me, because I want to build a bit of a consortium and a group around that. And I need lab scientists.
Perfect. I should also mention that Richard has written a piece for the December issue of the Biomedical Scientist, all covering anti-coagulant reversal agents, and it's a fascinating read. So if you'd enjoyed this discussion, then please do go pick up the magazine and have a read. Richard, thank you so much for joining us. It's been an absolute pleasure. Thanks to Richard for taking part in this episode, and thank you so much for listening. I hope you enjoyed the podcast, and remember that this episode can be used as reflective learning for your CPD. Please do keep an eye on the IBMS social media feeds and website for future episodes, and thanks again for listening.